please join me in welcoming Professor De Kretzer.
David, you might have to turn around to further. Got a screen.
We have a screen.
No, it's not. David. I've got the names wrong.
Thank you so much.
We'll get better to do it now. All right, here we go.
Well, thank you very much for the opportunity to talk with you, and I'd like to tell you a little bit about a journey which basically took me on a journey where I didn't expect to go. And basically research is such that you never know where it will take you. And I started off, I'm a reproductive endocrinologist by training, meaning I deal with things like hormones that control fertility, and in particular in relationship to men who had problems with infertility. But you'll see that this journey has been quite interesting. But again, it's interesting how you change your pathway. I'm a medical graduate, I was going to be a surgeon, and I basically took some time out to do an MD by thesis. And during that time I was looking around for projects, and I met a man called Pinkus Taft, who was a fantastic endocrinologist at the Alfred Hospital. And basically I was looking around for projects, and he found one for me which was in endocrinology. And basically I was hooked after that, and one of the things he taught me about was how to express complex ideas to patients that he would have to deal with. He was a master of communication. Becoming an endocrinologist meant that I needed training, and the options were, I was fortunate to get a National Institutes of Health fellowship from the USA. So I actually went to Seattle, the University of Washington, and had 2 years there to learn some new techniques. And basically that started me on a journey to, if you like, prove something that had been claimed to exist in 1932. And I'll come back to that in a minute. So I'm going to be talking on the slide here about the pituitary gland. That's that red structure that sits just hanging from the base of your brain. It's the master controller of hormones. And if you put your fingers like that, and where they meet is roughly where your pituitary gland is, okay? It's right in there behind your eyes. And if you have a tumor of your pituitary gland, it presses on the nerves that interfere with vision, and that's sometimes the first thing that somebody knows that they actually have. So that's the organ, if you like, that functions. I'm going to tell you about a thing called the inhibin hypothesis. Now, the ovary and the testis are controlled by hormones that come from the pituitary gland. So they send signals out to stimulate sperm and egg production, and the ovary in return sends back a signal to say, hey, enough stimulation or not enough stimulation. And hormones such as estrogen and testosterone are part of that. We call it a negative feedback system. As they go up, the stimulus from the pituitary comes down. Now, one of the issues really had been that there's a molecule called follicle-stimulating hormone. It stimulates the little sacs in which eggs in the ovary develop. And the hormone from the pituitary that stimulates that is called follicle-stimulating hormone. But people didn't know what the negative feedback was that controlled FSH. Some people suggested it was testosterone or estrogen, and they do actually play a role in the control, negative control. But there was evidence occurring and put together really a long time ago by a man called McCulloch in 1932, who said there was a putative hormone called inhibin that existed. Now, nobody had isolated inhibin. When I came back from America, we started to look to see if we could prove that this was a mythical substance or whether it was real. It took us from 1972 to 1984 to finally isolate and purify that hormone. Today, with the technologies available, you could do it in 6 months or less. So things have changed a lot, but there it is. So it took us on a journey. Basically, inhibin basically suppresses FSH. There's another molecule called Activin, which stimulates FSH. Sorry for that. And there's a third molecule called Follistatin, which also suppresses FSH. Now, how are these related? Well, down the bottom of the slide here, you'll see that inhibin, and don't worry about the A and B, let's just talk about inhibin. It's made up of 2 parts, an alpha and a beta subunit, which are joined together. If 2 betas join together and there's no alpha, you make activin, which stimulates FSH. An interesting system. So the alpha, beta suppresses it, that's inhibin. So we isolated inhibin, we isolated its function, and then shortly after, people found activin, which actually stimulates FSH. And then this third molecule, follistatin, which suppresses FSH, works by grabbing activin and stopping it functioning. So if activin, which stimulates FSH, is grabbed and stops it functioning, FSH levels go down. So I want to talk to you about, so these were hormones that were controlling the pituitary gland and the crosstalk between the ovary and the testes. But what's surprising is that it took us on a totally different journey, which I'd like to describe to you. I'm going to talk mostly about Activin A. There are actually other types of Activin, Activin B and Activin AB, but let's not worry about those and just concentrate on Activin A. They're part of a bigger family of proteins called the transforming growth factor beta superfamily. And these are proteins, and they, when you say they belong to a family, it means their structures are very similar. They're, if you like, modifications on a theme. And they all have different functions, usually locally in terms of different organ systems and different processes, such as inflammation. And as I said, follistatin stops activin functioning by grabbing it and basically targeting it to cells that cause the degradation of that complex. How it does that really doesn't matter. So basically, one of the interesting things about activin is if you look at its structure, activin's a protein, so it's made up of subunits called amino acids. And if you looked at the amino acid sequence, which is what identifies that protein, that's the sequence. And activin is basically 100% identical in man, in the cow, in the pig, in the rabbit, and in the rat. It's 99% identical to what's in the sheep, and it's 97% identical, even 87% identical to a frog. So it tells you that the gene which actually encodes for that protein has been conserved during evolution. If some species fiddled around with that gene, they actually haven't survived. And follistatin, the molecule that blocks activin, is about 98%. So these 2 molecules tell me immediately they're very, very important in some way. Maybe in reproduction, so it's just the follicle. So basically, activin was also identified for several other actions down in the bottom 4 here. It stimulates red cell formation in the bone marrow, part of your blood forming system. It stimulates, during development, the formation of muscles. It survives, helps nerve cells survive. So it's expressed in the brain and actually is part of protecting nerve cells. And it affects some cells as part of the immune system. So how did we get into the inflammation area? Well, when we isolated inhibin to prove the hypothesis, if inhibin was coming from the ovary or the testis, if you castrated the animal, it should disappear from the circulation. It did just that. We just simply took some animals, castrated them, measured the inhibin levels, and they disappeared. So it told us immediately that was the factory where it was produced. So when we identified follistatin, we said, let's do the same experiment and see what actually happened. Well, here we actually ran into some interesting data. So that's what happens if you take a sheep and we sampled over a period of really 24 hours or longer, just blood samples, and we basically measured follistatin, and you can see it doesn't really change. Well, we castrated the animals, and instead of disappearing, follistatin went up. we were quite surprised by this. But whenever you run an experiment, you always have a control. And you actually anesthetize the animals, operate on them, but you don't castrate them. You just sew up the scrotum of the liver. And you can see on this slide that follistatin went up under those circumstances, which told us immediately this was part of the body's reaction to surgery, to surgical stress, or maybe to inflammation. Probably not inflammation because it's a very short period of time. You don't get an inflammatory response within 24 hours. So this started us thinking about it. Now at that particular time, we couldn't measure activin. We didn't have the technique to do it. And we put away these samples to go back and measure activin when we could. But sometimes things happen in the laboratory. You had a freezer disaster. Instead of freezing it, heated. And that was the end of the sample. So basically we had to start again. Oops, lost it. Okay. So that started us to develop the techniques to measure activin, which we finally did, and then to actually see what would happen under those circumstances with activin. And basically, what we started to do then was to show that activin, like follistatin, responds to things like surgical stress, to inflammation, and this took us on a very interesting journey. I'm not quite sure what happened. I promise I didn't touch it. Here we go. So basically, to keep the story going, we then decided, let's look at inflammation. Now, there's a very simple way you can eat inflammation. You know that if you eat a bad corn, within about an hour or two, you start to feel shivery and shaky, and you start to vomit. And that's part of the body's reaction to that basically inflammatory stimulation. Now, to mimic a bad corn, what you can do is you take bacteria called salmonella, which cause gastroenteritis, okay? And you take the outer wall of the bacteria off, and you actually create a preparation called lipopolysaccharide. We call it LPS as a short form. So lipopolysaccharide is a way of, if you like, artificially stimulating an inflammatory reaction. So what we did then was to take sheep initially and give an inflammatory, give them a lipopolysaccharide injection. And what we found was that with that stimulus, we raised the levels of activin very rapidly. So activin was responding to an inflammatory stimulus. Now, sheep are expensive animals to deal with. They have one advantage: you can take a lot of blood from them, blood samples. But in the end, what we did was to switch to the usual laboratory animal that we use, which is a mouse. And when you get it back, what I'd like to do is to show you what actually happens to a number of proteins which are part of your inflammatory response, including activin, when you give, if you like, that challenge with lipopolysaccharide, mimicking you eating a bad corn. And this starts to become very, very interesting, because within here's so there's basically so there's the stimulus that's occurring with castration. Now here's the sheep studies. That's the temperature rise when you give a lipopolysaccharide. So you can see that in 45 minutes, the temperature in the sheep has risen. There's what actually happens to activin. Very, very rapid turn on. And there's tumor necrosis factor alpha, which is one of the molecules that is well known to be part of the inflammatory response. If you have inflammatory bowel disease, there are antibodies or medicines now that actually stop TNF-alpha working as a therapy for inflammatory bowel disease. So activin is released very early. By itself, it doesn't actually stimulate a temperature. It's not affected by blocking other molecules called prostaglandins. And basically, we started to become very interested in this molecule. So here's now the data in mice. Here's the activin levels. You can see within an hour you get a very large rise. The levels actually continue to be elevated almost for 24 hours after that single challenge with lipopolysaccharide. You can see that follistatin rises after this spike of activin. And basically what happens is that activin stimulates its binding protein follistatin. It's a safety mechanism, because if I was to infuse you with activin, it will knock off your liver cells and will knock off your lymphocytes, which are part of your body's response to immune challenges. So it's a dangerous molecule that's very important. It needs to be controlled. But if it's out of control, it's bad news. It kills you, effectively. So here's the summary data. Here's the rise that actually happens with activin. Here's the rise of follistatin. And here's the rise you get of TNF-alpha. This is a marker of the inflammatory response. The response with IL-6, which is shown, just concentrate on the not coloured area here. There's IL-6, and here's IL-1. Now, the second set of coloured data there is we gave these mice just a microgram of follistatin before, half an hour before we gave the lipopolysaccharide challenge to inflammate. So that activin is now coming out in the presence of its binding protein, which stops it acting. So what you can see is when you did that, we didn't get this big rise of follistatin, immediately telling us that activin was stimulating follistatin. We halved the TNF-alpha response. We changed the IL-6 response. Instead of coming out with a flat peak here, it now comes out much earlier and it's augmented. And with IL-1, we saw movement backwards, but we didn't get the augmentation. So blocking the actions of activin changed the whole cytokine response, inflammatory response. So we believed we were really onto something that was very, very important. Now, skip that one. Now clearly, if you give enough lipopolysaccharide, you'll kill the mouse. Okay? And we did this. It's an experiment which we need to do. And you can see that basically here's the mortality that occurs. These are percent survival of mice which have given a big dose of lipopolysaccharide. And here's what actually happens when we now give 10 micrograms of follistatin, a larger dose. You see you halve the mortality. So blocking that rise of activin halves the mortality in a model of, if you like, a profound inflammatory response. So can we translate that into human studies? So here's actually the data, the activin levels on the mice that died and the activin levels on the mice that survived, okay? So the mice that died had much higher levels of activin. Now you can translate that into human data. So here's a patient in intensive care with septicaemia. And you can see the red markers, a thing called CRP, which is a commonly used marker. If you go into hospital with an inflammatory disease, they measure your CRP. It's a marker of inflammation. And this particular patient's in hospital over 26 days in ICU. And you can see that you get rises of activin and follistatin in concordance with the CRP levels when you get marked inflammation. And basically here's some study from a collection of a series of patients. And the black skulls indicate that the patients died, okay? And the nice smiling faces of the patients who survived. And take my word for it, the ones who die have higher levels of activin, okay? So if you measure this in somebody who's in intensive care, it tells you to some extent whether they're going to live or die. Let's see that one. What about in patients with an inflammatory disease called rheumatoid arthritis? So taking fluid out of the joints with people with rheumatoid arthritis, here's the activin levels in yellow. If you have osteoarthritis, which is a degenerative disease but not inflammatory, you can see the activin levels are a bit elevated, but not to the same extent as an inflammatory disease. So again, here's an inflammatory disease, activin is elevated. If you take patients with heart failure and classified them according to what's called the New York Heart criteria. The higher the score down the bottom here, up to 4, that's the worse your heart failure. And you can see that activin levels rise as part of heart failure as well. If you take patients, or take a model of a myocardial infarction, a heart attack, by tying off the artery on a particular part of the heart, and creating, if you like, damage to the heart muscle, inflammation that takes place. And again, what actually happens is that you get inflammatory cells coming into into the scar formation, and basically activin increases. And activin increases, but it also stimulates the process of scar formation. So it's an inflammatory stimulus, but also starts the process of healing by stimulating, if you like, the healing process of a scar. You can take another model, which is if you give a rat carbon tetrachloride, it causes cirrhosis of the liver, causes an inflammatory response. And again, basically that's a model that we use to look at activin in hepatic cirrhosis. And you can see again, in the animals treated with carbon tetrachloride, the activin levels are higher than the animals which actually receive follistatin as a method of actually blocking the actions of activin and decreasing the levels of cirrhosis. So we're starting to see the potential of follistatin emerging as a potential therapeutic for anti-inflammatory and anti-fibrotic conditions. And you can do the same thing in Bleomycin is an anti-cancer drug, and basically it sometimes causes lung fibrosis, and activin is shown to be elevated in a mouse model of Bleomycin-induced fibrosis. And if you give follistatin, it blocks that fibrosis occurring. It blocks the inflammatory response. The same thing can be shown in skin. Now what you can do in a mouse is to create a mouse, for instance, where activin is strongly expressed in the skin. And if that actually happens, the mouse's wound, when you wound it, heals very quickly with a very significant scar. If you make another mouse which actually expresses follistatin in the skin, and then you wound that mouse, the healing is slower, but there's no scar formation. So again, you can see potentials there for follistatin in various types of repressing scar formation. And if you take wound healing, basically a very nice study out of Switzerland showed that follistatin and activin are expressed in basically wounds and expressed particularly in the lining of blood vessels. If you turn to inflammatory bowel disease, there was a study from Japan, and don't worry about the details, which actually used several different ways of inducing inflammatory bowel disease in mice. And what you can see down the bottom is that activin levels are elevated in the yellow, and basically follistatin is elevated. And again, in the mice that were treated with follistatin, you get a survival rate. You can see down the bottom here, there's a survival curve. The green is the mice that were with inflammatory bowel disease that were treated with follistatin, and here's the death curve with mice that weren't. And you can translate that into humans as well. Now what about burns injuries? So here are basically a number of patients where they've had burns, severe burns injuries, and we're measuring now activin and follistatin in the blood of these patients. The dotted line here is the normal upper limit of normal. And you can see that in all of these patients with burns injuries, the activin levels and the follistatin levels are elevated. Again, burn injury is a very profound inflammatory stimulus. You know that you get a blister forming very quickly after you've had a burn. That's part of your inflammatory response. And if you look at that blister fluid, you see that the blister fluid actually has very high. Remember, we're down here when you talk about what normal levels are in the bloodstream, 0.1. You're going quite significantly elevated. And here's, if you like, the expression of basically activin in the edge of the burns injury in a patient with burns injuries, and also follistatin being expressed here. So these are players that are important. I won't bother with that, doesn't show up very well. So summarising all of that, we've shown that activin is expressed in cardiac muscle cells during ischemic injury. We've shown that activin is produced in the liver during injury and scar formation. We've shown that follistatin can block fibrosis that occurs in the lung, and also that it's important in wound injuries and in burns. And there's a thing called keloids. Best example is my plastic surgeon colleague Graham Southwick has a patient who basically had his ears pierced. 3 months later he had the size of walnuts hanging there. Graham's now done 6 operations on this man, removes it beautifully, 6 months later it's sitting there. So he's a real potential candidate to use follistatin to block that process, but we still can't use it in humans yet. So basically we're talking now about some very interesting molecules. So just to come back to that pro-inflammatory action, okay, about inflammatory bowel disease, I want to show you some data which I'll just... One of the things we had to do once we got into human work was to get normal ranges. Absolutely essential. So you have to find maybe 100 normal people, men and women of different ages, with no injury, no illness, and draw blood from them and then measure the normal range. Because that's what you're going to be comparing things to. So we actually now have blood samples which are stored so that if our assay changes, we can recalibrate it and go back and continue to do that. So effectively, we've now got normal ranges for these patients. And I want to tell you about a study which was done in conjunction with a colleague in Finland. He had 518 patients who were part of a study of 958 patients who were admitted consecutively to intensive care units around Finland with acute respiratory failure. And we were fortunate to get blood samples from those patients. Not in all of them, but enough to do a very interesting study. So basically, of these 518 patients, some of them had what's called acute respiratory distress syndrome, meaning you're very, very, very ill. Some of them had an acute lung injury. And basically, they were all seriously ill and took them into intensive care. Now, acute respiratory failure can arise from cardiovascular disease, it can arise from gastrointestinal disease, it can arise from sepsis, it can arise from metabolic disorders, and obviously acute respiratory failure. So there's a mixture of that. So here's our normal range data. You can see that that's the activin A levels that are up there in men and in women, and activin B and the follistatin levels here. And once you get above up this area here, that's outside the normal range. And so when you start to look at patients with those conditions in acute respiratory failure, it doesn't show up very well, but the black bar at the start of each of these graphs is the normal range, okay? And you can see that the patients in these studies with a variety of causes all have elevated levels of activin, activin A and activin B. Now basically I have a young colleague who is very good at analysing data and coming out with, can you make predictions on what you measure? So here's the survival data that we can predict from that study. So basically, if your activin A and B levels are in the normal range, your survival, and these are blood samples drawn in your first 5 days in intensive care, and we're looking at survival up to 1 year or more after your ICU admission. So we can predict with 80% accuracy that if your activin A and B levels are normal, that's your survival curve up the top here. Pretty good. If both of them are elevated, you're down here, and that's bad, okay? So immediately it tells us that these molecules are very important markers and that we need to start to work to actually use follistatin to block their negative actions. And so basically you can actually model this even further, and you can come up with survival curves which are differently stratified depending on whether your activin A and B are markedly elevated or lower. And obviously, this is your top survival curve. If you're down here, it's not really very, very good at all. So why do people die if their activin A and B levels? I've told you about the liver cells being damaged. I've told you about, if you like, the lymphocytes, which are part of the immune system, being damaged and destroyed. Activin also causes the release of nitric oxide of blood vessels, and that actually can cause inflammation and cause actually changes in the way your blood vessels function. So, sitting here are the changes that occur, elevated levels of activin, stimulating the inflammatory response, driving the process to scar formation. And again, to attenuate that scar, you can use follistatin. So we think this is a very important thing. We now have a small company that's actually manufacturing follistatin. Fortunately now we can do it in Australia, which becomes very important because there's a facility up in Brisbane. And you start off with 1 litre. We're now into 1,000 litre fermentations. One of those 1,000 litre fermentations costs about $250,000 to do. But we're now manufacturing large amounts of follistatin. The next step is to actually do toxicity testing, just in mice and then in primates. And then hopefully by middle of next year, providing everything goes okay, we hope to be in trials in patients with cystic fibrosis. Delivering follistatin through a nebulizer system. We've shown that you can actually get a spray, just like you take an asthma spray. The nebulizing process doesn't destroy follistatin, so it's a good way of delivering it for patients who have lung conditions. And so we hope to be in trials in cystic fibrosis in that area. But there's a myriad of applications. Applications which don't involve even giving it to patients. For instance, if you take a lung in Sydney for transplant, send it down to Melbourne, it's off its blood supply for 8 or 9 hours. When you put blood back into that lung, you create an inflammatory response. So that's damaging the transplant before you even put it in. Just to put follistatin into the preservation solution, we've done this experiment in dog lungs, we can show quite clearly the quality of the lung that you preserve is far, far superior. And again, that means we don't actually have to give it to a patient, we're just simply putting it into the preservation solution. And obviously when you're developing something, it's nice to have some applications which don't involve giving it to patients, because one of the things that happens is that you start off with 10 normal people to give follistatin to. That's the first thing you do. Then you give it to maybe 10 patients with cystic fibrosis. Everything goes well, they do well. Then you get out to 100 patients, and 2 of them develop some side effect. Real trouble, okay? So to have a backup where you can use it for something that doesn't involve giving it to patients is, to some extent, when you're running a company which is actually using other people's money, it's nice to be able to give them some assurance that there is this possibility. So there's a journey which starts off in reproduction. I could never have predicted that I would have been working here. And this is the sort of thing we're trying to get across to politicians, that research and innovation are critical. You know, we have a potential therapeutic. Hopefully we can try and do most of the development in Australia. At least we can manufacture in Australia now. We still can't, we don't have a testing facility for animal testing to do the toxicology in Australia. We're going to have to send our material to Canada to do that. But that's a story which I hope you find interesting. I'm happy to answer questions.
Septic arthritis, how does this fit into the scene that you've described?
Septic arthritis? Well, septic arthritis is an inflammation. It's an infection of your joint, and effectively, just like I showed the data with rheumatoid arthritis, you will get secretion of activin and follistatin, activin particularly in the inflammatory phase in that particular joint. So yeah, it's there's potential there. you have to recognise that part of your inflammatory response is a healing response, so you don't want to shut it down completely. And one of the concerns I have is that, okay, we've got people on long-term follistatin treatment. You know, what actually happens if they get an infection? We need to be able to stop the follistatin. Well, fortunately, follistatin gets removed from the circulation within about 24 hours of that. There's an alternative way of blocking activin. Activin to act has to be detected by receptors, and the receptor then triggers a response in the cell. You can make what we call a soluble activin receptor, which acts like a receptor, but it doesn't have the bit that triggers the cell to respond. So there are soluble activin receptor blockers which are available as well. But they tend to have a longer half-life. They last for about 8 days. So basically, follistatin has the advantage that you can remove it and it disappears in 24 hours or so.
George?
David, George Martin, Biochemistry from Melbourne University. There's been some epidemiological studies done in India about curcumin, which is, as we know, the active ingredient in turmeric and curries. And that seems to have sort of an inhibitory effect on activin. Do you have any thoughts in this area as opposed to just follistatin as an inhibitor to activin in inflammatory situations?
So there's some situations where activin is
Yeah,
suppressed.
curcumin is used. And there's 10,000 people that have been tested in India and found out that curcumin is very active in this area. It's a very natural product, so there's no side effects to test. And I just thought whether you come across that study.
No, I haven't come across that, but I'll certainly follow it up.
Because it's an interesting point in terms of any inflammation. People that take curcumin is found to have the inflammatory effect, and you might have an inhibitory effect as a different model.
Did everybody hear that question over here?
David, I work in spinal cord injury at the Austin Hospital. I'm interested in the applicability of this to people who have pressure sore injuries because they can't move like you and I do when we feel like we need to change our position. They stay in hospital for months and months. They can't mobilise or do anything. Just interested in any potential applicability to make the wound healing faster. Often goes right through to their buttock bones.
That's an interesting one. Certainly, I mean, you could cut the inflammation by using topical follistatin once it's available. But you still want some degree of inflammation to process to heal. So, you know, it's a balancing act, I would think. You know. These are the things that, until we get to follistatin, I mean, quite clearly the mouse studies and so on, and you know, we've injected follistatin into making injuries and looking at the healing process. And one of the good things about follistatin, when you inject it into the skin or to a region, it doesn't diffuse very far because it tends to attach to the surfaces of cells in the vicinity. So it doesn't, you know, if you put something into the wound on the back of the mouse, it doesn't get into the blood circulation and so on. So it has that effect locally.
Any other questions?
Well, one thing, David, it would be interesting to me to know about you come to a person like senior people too, and they've been happy to speak to you occasionally, and not necessarily on medical terms. I must tell you, I'm not complaining about your medical terms. I did understand them, and if I understood them fully, I would certainly be sublime to you, but I couldn't understand them fully. I know that medically you've got to speak. You can only do that well. And I'd invite you to one of my luncheons, which I haven't indeed done that far, but certainly get this sort of thing off the screen if you want. But I don't know whether you want it. But if you want things like that, you can put it through to these people.
Thank you, Chris.
Is it something else, or is it good?
Thank you, Chris.
I've been talking on a range of topics.
Thank you, Chris. Dr. McCready, you can answer a question about
Oh, a fairly naive question about relationship between activin levels and C-reactive protein levels, and do they correlate very well, and could the one replace another?
They do correlate very well. There's no question about that. Yes, the active proteins are the good marker of inflammatory processes. I guess the activin tells you like one of the substances that drive that inflammatory reaction. They're not exclusive. You know,
It'd be a much more expensive test, I presume, and I can't imagine it would replace CRP.
I would have no doubt it would replace CRP. Well, I don't think you need to replace it. It would be something you would do jointly. No, I mean basically the assay systems are going to be already systems that not allowed.
I have a question about development. So you're talking about maybe, some sort of preservation solution, infiltration with the statin. And back to that question about wounds, is it a shorter development time for topical as opposed to as ingested or injected therapy? Is it, or is it a longer time?
Look, it's pretty much the same,
Oh,
in that you actually have to take the molecule you're developing through a process, do the toxicity testing to get it approved.
Okay.
We have given on grounds with the support of Cabrini ethics to one patient. He was a young man who got basically about 70% burns of his body. Graham Southwick was the doctor who looked after him. Basically, he grafted him, and now it's a couple of years after that, the grafts are starting to scar, and so he was having difficulty moving his head and even opening his mouth. So Graham managed to get Cabrini's permission to actually use reagent-grade follistatin in this patient. The scarring was so thick that you actually couldn't get a needle in under the scar. So what we had to do was Graham operated on him and made a couple of incisions about 2 weeks before to free up the scar tissue, and then we put in follistatin. He can do that on that side, but he can't do it on the other. Now that to me was beyond my expectations. There's no question that if you block an acute inflammatory response that's proceeding to a scar, you will decrease the scar. But to take an old scar and to do that was quite surprising. But that fits in with some data that's starting to emerge that a scar just isn't a dead tissue, that it is actually remodeling all the time, and that there's some scar tissue that's being reabsorbed and other that's being laid down. So.
I think we won't conclude questions, sorry.
Sure. Now, you were the government for 5 years.
I have to come over there.
So,
The hearing aid doesn't work very well.
I know it's limited, but you were government for 5 years. To what extent did that work in the Serum?
Okay, so the question was, did governorship interrupt my work? It did a bit. Governorship has a bit of free time, so one's engaging with things on email that makes something useful. There were once a fortnight I would spend 3 hours out at the lab with my younger colleagues, and occasionally they would come in and enjoy the hospitality of Government House around the table, which we have a very pleasant discussion. So it slowed some things down, but not to the point of removing my interest in you.